Good morning, everybody, and welcome to the North Carolina April Vineyards workshop. Today, we are talking about a lot of research related to plant pathogens in the next 45 minutes. My name is Mark Hoffman. I am the Small Fruits Extension Specialist at NC State University. And this presentation is recorded because I have to be in the field at the moment to harvest strawberries. However, I am on the phone on this call and will be able to answer questions later. The presentation is at the moment given by my PhD student, Kyle Friedman. And we are talking today a lot about two different diseases, systemic diseases, and one of them are grapevine viruses, which we did quite extensive work on in the last 3 years here in North Carolina. And then we also going to talk about grapevine trunk diseases, which we are working on since last year, and we continue to work on in the next 2 years. So first, please feel invited to talk a little bit about grapevine viruses. So now, as with every research project, it is not just one person who's doing the research, it's a whole team of people who do this research. And in this particular project on grapevine viruses, which was from 2017-18 till the end of 2020, we had quite a bit of people involved in this, with the exception. So I was involved in it as well. But I would like to highlight that my research assistant, Emma Volk, was involved in this and is involved in this quite a bit. And then also Win Talton from the micropropagation unit at NC State University and Christy Almeida from NC State University at the micropropagation unit. Without those two people, the whole grapevine virus project would not have been worked. So those are very, very, very important to be on board. We also involved Matt Bertone, who's the director of the Plant Disease and Insect Clinic at NC State, and we also got quite a bit of help from the FPS, from Maha Al-Ravini at UC Davis, and also from Sudi and the Plant Pathology Department of UC Davis, and also at UCAIRS. And Mitsuo Nita helped us a little bit as well in the beginning of this project. So again, a lot of this is collaborative, but as with every good research project, every research project has several objectives, and we had 3 objectives in our specific research here. The first one was to identify what viruses are here in North Carolina. The second one was to establish testing capacity for grapevine viruses, which was not established at the time when I came. And then the third one was also to establish a testing service, which is different from having the capacity for growers to use in North Carolina. So we did have 3 objectives here, and we're going to go over all those 3 and see what we did and how we did it and what's available to you now. So grapes can harbor quite a bit of viruses, and as we know all now from 2020, everybody can harbor viruses. Not all viruses can cause disease, but some can, and some can be very severe. And that's the same with grapevines, but some grapevine viruses can cause quite some severe disease symptoms. So the things which we see on the plant are disease symptoms, and the viruses are the pathogens which cause or are associated with those disease symptoms. And then some of them are very widespread, some of those viruses. And when I came in 2017, in the later half of 2017, in July 2017, I visited quite a bit of vineyards right away, and I saw a lot of disease symptoms which were very virus-like, but we did not know whether or not it is a virus, and then we also didn't know which virus we have here. And there are quite some surprises which come down the line. After those 3 years, we have a much better handling on which kind of viruses we know, and we also now can test for viruses. So the most common grapevine viruses are which cause disease and which are worldwide spread is grapevine leaf roll associated virus, There are several variants of those. We also call them GLRAV, 1 to 11. And then grapevine red blotch virus, GRBV, which is also very widespread in the world. And then two other viruses to watch are grapevine virus A and grapevine virus B. They're a little bit newer on the radar, but they can cause some disease symptoms as well. So if we talk about viruses, I just want to make sure that we all understand what we're talking about. Viruses are one category of a pathogen, but there are several other pathogens around which can attack grapevines. And pathogens are usually microorganisms, which we call microorganisms. It's a whole bunch of different kingdoms which are under those umbrella. Bacteria is one microorganism, and there are some bacterial pathogens which are pretty severe. Two of them, one of them would be Agrobacterium vitis, which is a bacterium that can cause crown gall, or several strains of this bacteria can cause crown gall. And then, of course, Xylella fastidiosa is another bacterium that causes Pierce's disease and can be transmitted through insects. But that is, again, a bacterium which is very different to a virus or to a fungal pathogen. So fungal pathogens are pathogens that are from the kingdom of fungi. They're very differently structured compared to bacteria, much more complex than bacteria, and much more widespread. In the grape world, at least, there are a lot of fungal pathogens which can attack grapes. Some of them are shown here, like downy mildew, or powdery mildew, or gray mold. Those are all fungal pathogens. And then also the trunk diseases, which we could talk in the second part of this presentation, are also fungal pathogens. So those are from the family of fungi, which is a very big, severe, large family with a lot of different morphotypes. And we will just talk about a few of them later in this talk, if we talk about wood-borne fungal pathogens or grapevine trunk diseases. So now we will talk about viral diseases, which is a third category of pathogens, and those are viruses. And I think after 2020 and after COVID-19, I think everyone should know at least what a virus is. We're still going to explain it, but of course, viruses can also affect any kind of different organism. A plant is an organism like we are as humans or animals. So there are animal viruses. There are animal viruses which can jump from animals on humans, like we saw with COVID. And there are also plant viruses. Thankfully, they usually don't jump on humans, but they can be still very severe for plants and can cause quite some damage on plants as well. So now this is like just a little overview, and I want to just show that because I showed this like 2 years ago in Surrey at the Surrey Community College meeting. And at that point, we did not know what the severity of those two diseases were in North Carolina. So we did our research, and now we know. And we're going to talk about this today. Just as an overview again, virus diseases are usually RNA or DNA particles. Trunk diseases are fungal organisms. They're fully functional organisms, while viruses are not fully functional organisms. Viruses are particles that need another organism to be functioning. So that's a big difference. A virus needs a functioning cell of a different host organism to be functioning. The whole group of viruses works like that. While fungi can be pathogenic and can use other organisms as host, but they don't have to. There are also other fungi which do not need other plants or organisms as a host. So now let's talk a little bit more about viruses, grapevine leaf roll associated viruses in particular. So as I said, those are one of the most common grapevine viruses are the grapevine leaf roll associated viruses. There are currently 11 variants. They're clustered into 4 different clades genetically. And those viruses, all of those viruses are associated with what we see later in the field, grapevine leaf roll disease. So the disease symptoms of grapevine leaf roll, those are associated with some or one of those variants. So you can see here a picture of an electron microscope of an actual grapevine leaf roll virus 3. That's the variant 3. Those are very complex viruses. They're RNA viruses, very large RNA viruses, 13 to 19,000 nucleotides large, depends on the variant. And they have a protein hull, so they're relatively complex viruses. They're around for a long time. The disease symptoms were first described, several hundred years ago from Europe, before grape phylloxera was introduced in Europe. And we know that they spread worldwide. There are reports from the early 20s from the United States which already describe similar disease symptoms. And they're transmitted by scale insects and mealybugs. Most of the variants are proven to be transmitted by those insects. Mealybugs can have different vector capacity, but we're not going to talk about this part today. But there are several mealybug species, and not each species is transmitting the virus with the same capacity. The most common viruses which we find in the United States are leaf roll associated virus 2, 3, 4, and 7. So those are the most common variants, but again, all the other variants are detected in the United States as well. So now, if we look at disease symptoms, this is a very beautiful picture. If we look at disease symptoms, we see usually green inner veins and red cultivars. So this is in red. This is a Merlot in the Yadkin Valley, in red cultivars, and we do see red interveins often at the end of the season when the virus titers get high. That's the same for red blotch virus. Usually around harvest or after harvest, that is when you would see those patterns emerge. So now, those virus symptoms don't have to look like this. As you can see here, they can have all kinds of different forms. On the left-hand side, you see that's a very similar form that I just showed with the green inner ways and the red with the green veins and the red inner veins. But you see also here in the middle that it can be fully red, and you see a lot of curling of leaves, especially on the right-hand side. That's very typical. If you see those curling, that's very typical for a leaf roll associated for leaf roll disease. So now, on white cultivars, this is much more difficult to detect just by the looks. It often looks like chlorosis or some nutrition problem. You can see here but you have a little bit of leaf rolling going on, especially in the lower picture. You can see that as well. But you often have like those yellow spots, a little bit crispy leaves. That can all be a symptom for virus as well. Impact on fruit and wine health is quite severe. It can lead to declined wine health, so to like less longevity of a vineyard at the end. You will have declined fruit quality in a leaf roll diseased vineyard, and also your yield will be declining over time. So impact can be quite severe. It's not necessarily a direct dieback, but you will see a lot of stunted growth and less vigorous vines and less fruitful vines, and also lower fruit quality if you don't manage those diseases in your vineyard. So now the second virus cluster we were looking at was grapevine red blotch virus. So that's a very different virus than the leaf roll. Leaf roll virus, as I said, is a very big, is an RNA virus, has a lot of nucleotides, has a protein hull around it, so it's a very complex virus, while the red blotch virus is a very small virus, and it's not an RNA virus, it's a DNA virus. So it's basically what is in our cells at in the core, and it has a relatively small, between 3,100 and 3,200 nucleotides small. You can see here, that is how it looks like. And the viruses can cause grapevine red blotch disease. So that is what we see. Disease symptoms are very similar to leaf roll disease symptoms. The virus is completely different. It's a completely different virus. That is relatively new. It's discovered 2008 in California in a vineyard which showed leaf roll symptoms, but couldn't test. They couldn't test for leaf roll there, so they found a new virus. Since then, it has been discovered in quite a bit of corners in the world. We also know that there is one vector which can transmit the virus, which is a three-cornered alfalfa hopper. How effective that vector is in transmitting the virus, that is a second question which we do not know. And we also don't know if there are other vectors which we probably have missed so far in research that can vector this virus as well. So there's a lot of research questions attached to this particular virus, which we didn't answer in our research project, but we hopefully can answer sometime in the future. So typically you would see redder veins, and the inner veins would remain green for like a while, but often they can be blotchy, and it is hard to tell apart those disease symptoms. As you can see here, the blotchiness you can totally see in the lower picture, but you also can see that they have green inner veins. Still, this wine was tested positive for red blotch and not for leaf roll. And you can see here again on the right hand side, that's a younger wine, also was tested positive for red blotch, but not for leaf roll. The symptoms are pretty similar. You will have declined wine health, fruit quality, also declined yield. So there is also a virus which needs to be managed if you have it in the vineyard. So now, management-wise, it is important to understand how much the severity is of this virus in North Carolina. That was one of the questions we wanted to answer. So we wanted to know what is actually here in North Carolina, what is the most common grapevine viruses we have, so that we get a better handle on what is more severe, what do we need to manage. And we thought, really honestly, my thought was, well, probably leaf roll is the most common one, because that is very common in other areas of the world. So we tested for quite a bit of leaf roll associated viruses, especially for variants 2, 3, 4, and 7, which we know are the most common ones in the United States. We also tested for red blotch viruses, and we looked in grapevine virus A and grapevine virus B. So that was the kind of core suite of like viruses which we tested for during our survey. And then we sampled 280 wines across 3 AVAs in Yadkin Valley, in Hendersonville, Blue Ridge AVA, and the Upper Hiawassee Highlands AVA. So those were the 3 AVAs. We looked at 2 different other counties outside those AVAs as well, one directly at the coast and one in the northeastern part of North Carolina. Those samples were taken always in like a 3-week window, often the last week in October and the first 2 weeks in November, so shortly before defoliation, when we saw symptoms, and that was done in 3 years, 2018, 19, and 20. And then and we only looked for red cultivars that showed symptoms, so that had red leaves or had some of the symptoms which I showed you. We made sure that our sampling was sterile, so it took us a while. We had to go with an ethanol bottle and different gloves and different bags, and made sure that all our samples are on ice so that we can ship them to the facility which would extract the RNA out of those samples. So we thank you to not just to Emma, who helped a lot sampling, but also to Karen Blado from Henderson County, Hannah Lepsch and Ellie Snyder in the Yadkin Valley, who helped us sampling a lot. So those samples were sent to the micropropagation unit at NC State, and then they extracted RNA. Emma also helped Win to extract RNA there. That RNA was then put into the freezer and then tested for all those viruses over time in the following months. So that is what happened, and our results are quite interesting. First of all, we looked out of those viruses we tested, we found leaf roll associated virus 3 and leaf roll associated virus 2 in in 5 counties where we sampled. Very high prevalence in Henderson County and Polk County, and then lower in Wilkes and Yadkin. And we found also some UFO-associated virus in Currituck County. However, if we look at red blotch virus, we found quite a bit of red blotch virus in almost every county we were looking at, just not in the Hiawassee region and not in Currituck. So if we look at the preservance of those viruses, so if you look at how much of the samples were actually tested positive, it is very clear that out of all samples which we took, grape red blotch virus was the one which was in 35% of all wines which we tested were positive for red blotch associated virus, while only 7% of all wines were tested positive for grapevine virus 3, leaf roll associated virus 3, and only 6.8% were positive for leaf roll associated virus 2. And just as a gimmick, we didn't look for Pierce's disease symptoms at all. We actually kind of avoided plants which had Pierce's disease symptoms if we saw them, but we still found 11% of the wines were positive also for Pierce's disease, although we did not necessarily look for Pierce's disease symptoms. But we didn't have a lot of cross-infection. Most of the wines were either positive for red blotch or positive for leaf roll, with the exception, I believe, of 2 vines which we tested. And also there was not a lot of cross-infection between Xylella and so Pierce's disease and any other virus. So as a result of this, we know now that red blotch is like one of the largest, is one of the viruses which we have to watch a lot in North Carolina, and that is a very good result for us to find new project funding to basically see if we can manage red blotch virus here in North Carolina, and maybe also to look what is actually transmitting it here in North Carolina. So if you look at the second objective of our study, we not just wanted to look in what viruses are here, but also we wanted to establish some testing capacity at NC State University. And we were very lucky here because we could work with the North Carolina Micropropagation and Repository Unit. They are also part of the Clean Plant Network, and they have berry responsibilities, and grapes do fall under the berry responsibilities. And they usually make sure that they have plants in tissue culture, which they then can sell to nurseries for them to start with clean plants. They're part of the Clean Plant Network. That is what they do. And at the moment, the FPS at UC Davis is also part of the Clean Plant Network, and probably the largest facility inside the Clean Plant Network. And we were lucky enough that we could work with them together to establish those grape virus testing capacity, because it was one of their internal aims as well. And we worked a lot with Wynn Tarleton. He's a research assistant at the NCSU Micropropagation and Repository, and the person to talk to if it comes down to virus testing and RNA extraction. And then Christy Almeida, who is the director of the NCSU and the MPRU. And we got quite a bit of support from the FPS at UC Davis as well. So they wanted us to establish those protocols as well. So without their help, we would not be able to do what we have done in the last 3 years. We also started to establish quite early, like the idea of entertaining a fee-based structure, not just to clean up plant material for nurseries, but also to test samples that come in from growers, which is very different. And we could establish a sample structure for in-state samples. And we're going to go over this in a second. But compared to most other virus fees, this is relatively cheap. So as I said, with the help of the Micropropagation and Repository Unit at the NC State University, we could develop some core capacities in virus testing of grape plants. And we're now able to test for leaf roll viruses, for red blotch, for virus A, virus B, and also Pierce's disease. So now, after we established this capacity, we also wanted to establish a testing service for growers. Again, that was in collaboration with Matt Bertone, who is the NC State director of the NC State Plant Disease and Insect Clinic. And we established a protocol, how samples can be submitted, how samples need to be entered, and how they need to be shipped. And I'm going to go over this in a second. But it is a relatively cumbersome process, but we need to make sure that the tissue is still having the virus, and especially because we're looking for a couple of RNA viruses, it can be very difficult to isolate them if the tissue is not handled in the correct way. So we did that not just for grapes. The micropropagation unit and the PDIC is working on that for all kinds of other fruit crops as well. But now we're looking especially on the service for grapes. And so usually you would first establish what if you have a viral symptom or not. And then you would collect your samples according to a protocol, which I will share with you after this slide. And then what is very important for you to know is that you cannot send one sample and expect it to be tested for several things. So if you want to send a sample for virus testing, that sample is solely and only used for virus sampling, and not to test for other pathogens as well. So we are going only from receiving a sample that will be picked up for the micropropagation unit, and then the micropropagation unit runs the panel of viruses that is requested by you, or the standard panel if you don't request anything. And then those results will be entered into the online system of the PDIC, where you can see your diagnosis. So that is how we work. If you want to have samples tested for other things than viruses, you will have to send in an extra sample. And usually we would run, for viruses, we would also run a Pierce's disease test if you not have tested for Pierce's disease before. The price for that is relatively low compared to most other virus testing facilities. So we have $50 for the RNA extraction, so for the actual processing of the sample, and then $30 for the handling of the sample. And then for each virus, we charge $15. So in a real world example, if you don't know what you have, but you describe the symptoms and your extension agent was there before with you, we would go through the most common viruses, which we now found out through our survey first, before we would charge for the whole suit. So basically, we would say, we would test for red blotch, leaf roll 3, leaf roll 2, and we would throw in an additional Pierce's disease test, and that would cost you $140 and shipping on ice. So, and that usually, in the most cases, you would have your answer after that. If everything is negative, then we would go deeper into all the other virus variants, which I showed you before, just to make sure that we know at the end whether or not it is a virus. So, again, talking about sampling, so this is important. We thought a lot about this, and what we do want to avoid is a cross-contamination of your sample. So it is very important that you sample a whole branch, and I'm trying to find the annotation. There you go. So what we would ask you to do is, for example, if you have a diseased plant, you would take a whole branch, but you cut it off here, and this is the only area where you touch it. You don't touch it anywhere else. You don't touch it here. You don't touch it here. You don't touch it here. And then you would take that part and basically cut this off, take this part, and then you enter everything in your at the PDIC, and try to get rid of this. There you go. So you enter your form in the PDIC, take your sample, touch it only at the base, put it into a plastic bag. Don't use a paper bag, use a plastic bag. Put into a plastic bag, put it on ice, not on dry ice, just cool it down overnight, and send it to the plant disease and insect clinic. So that is the protocol which you have to follow. Don't touch the samples on the leaves or on the petioles, especially. So you do not want to touch leaf and petioles, because that can cause cross contamination, and then we don't know if what we isolate from that is actually from that plant, or it might have come from another plant which you touched before. So you need to make sure that this is a relatively clean sample, and the best way to do is just take a whole branch, touch it at the base, and then put it into a bag and send it to us. That would be our way of like sampling. And again, and now you understand also why we don't want to test those for all kind of different pathogens. We're just only going to test that for viruses. All right. So now another thing, and just very quick, is another research project we're working on is grapevine trunk diseases. And again, I'm taking questions after this. I will be live for a couple of questions if you have questions about the sampling methods. But before we talk more about this, I want to touch base on the grapevine trunk disease, because that's another research project which we have. Again, a lot of people are involved in this, especially Sarah Villani and Rachel Kreis from NC State University. And we do a lot of that research here at Bernshire Vineyards. But there are many, many more who are involved in this trunk disease research. Grapevine trunk diseases are caused by a large variety of different fungal pathogens. Again, these are fungal pathogens, not viral pathogens. So now we're switching gears and we're going into a different disease, which is caused by a whole lot of different pathogens. And some of those pathogens are Eutypia dieback, which is a fungal pathogen caused by Eutypia lata, that's a fungi. And then Escargot disease complexes associated with a range of different fungal pathogens. And then Botrytis dieback are also associated with fungi from the Botrytis species. There are quite a bit of Botrytis species out there. So those are the three big groups of trunk diseases. And those trunk diseases are pretty severe. They can cause a lot of damage to the wine. They can and can enter the wine through several wounds in the wood, usually open wounds. Pruning wounds is one of the entry for fungal pathogens. Cold damage, especially if you get freeze injury and get cracks in trunk and cordon, that can cause severe damage, not just through the physical damage, but also through the entry of trunk diseases and trunk pathogens. And then, of course, other physical damage, like from a tractor or from trellis or etc., that again can cause entry wounds for pathogens. All those pathogens can also infect other plants, so we assume that there's always inoculum around. And rain and wet conditions do facilitate the spread. And since we are living here in a high precipitation area where we do have quite a bit of rainfall over the year, we assume that is a thriving factor for those trunk diseases to be more severe in our area than in the more dry Mediterranean climate for grapes. So disease symptoms are often those those black pies, which we see here, and if we do cuts through the cordon or through like larger permanent wood. If you then cut a cordon, as we did here, or a trunk into half, you can see that those trunk diseases then often can go through the whole area of the trunk. As you can see here on the right-hand side, see that black area is all infected. While on the left-hand side, you see here, if the entry wound was smaller and there was plenty of time to heal, we did not infect the main permanent structure here. You can see here, this is all not infected. So those are typical signs. From the outside, it looks, again, can look very similar. You see here is a mixed infection. You do have trunk diseases in here, but there's also some virus going on, as you can clearly see. On the right-hand side, that is a typical ESCA disease symptom, those tiger stripes, as we call them. Again, can be confused with a lot of different things, but we're going to go through this after this to like know how to identify those diseases. So now the question is, what do we need to research on those? There are a couple of questions we don't have to answer. For example, what to do after cold damage. First of all, you should manage your cold damage by selecting the correct site for your vineyard. That's very important. You will get a lot less cold damage if you grow in a good spot. And then the second is also cultivar selection. There's cultivars which are less cold hardy and cultivars which are more cold hardy. That's a big management tool for cold damage. Don't underestimate that. And then replacement and retraining of damaged vines is absolutely necessary. Don't leave cold damaged vines in your vineyard and don't attend the damage. You're going to have to retrain those vines, or even to replace if the cold damage went all the way to the grafting unit. So that is very, very important. If you do not do that, you facilitate the spread of trunk diseases, not just in that vine, but in your whole vineyard, because the vine just will be an inoculum for trunk diseases. So you will not be happy. And again, site selection is one of the most important parts to avoid cold damage. But again, research-wise, not a lot to do. Physical damage, also, there's not a lot of research we can do on this one. Basically, try to train your tractor drivers, be careful in the vineyard, make sure your rows are wide enough, make sure you have enough turnaround space. Again, vineyard and site establishment is a key to avoid physical damage to your vines. And also prune frequently, train your vines correctly so that you don't have any problem with your trellis system later. So again, that is a lot of education, but not a lot of research. But on pruning wood management, there is some good research done in Mediterranean and semi-arid climates, but we don't know if that works here. And we don't know what's the best method in North Carolina to attend to those diseases. We don't even know which kind of diseases we have in North Carolina, which trunk diseases are the most prevalent. And we started this last year in 2020. And as you all know, we got hit by frost and COVID was there. So 2020 was probably not the best year to start a research project. We did it anyways. And one of our key areas was to identify what is the most common pathogen. So what's our enemy here? What do we have in North Carolina? And then, of course, we wanted to start establishing some identification capacity also, so that we can use the PDIC, the Plant Disease and Insect Clinic, for you as a service center to get tested, to get your trunk diseases tested. And then also we wanted to look into best practices for pruning wood management in North Carolina. So in 2020, we started looking before COVID hit, and then we did get some extractions done, but then with COVID, everything shut down. And but we have found so far, and that is confirmed by studies which were done earlier, that we have mostly Botrytis furei species from wood samples with disease symptoms. We also found a lot of other things like Fusarium and Colletotrium, Neopestalopsiosis. But we also found quite some Botrytis furei species in North Carolina. And we also saw a lot of ESCA-related disease symptoms. We did not have the time to isolate anything from there so far, but we know we have ESCA because it has such distinct disease symptoms in North Carolina as well. We also did a small trial, which then was hit by frost. So those numbers are kind of not very telling. But I just want to let you know, we looked at delayed pruning as one of the methods, and also at the use of woody seal as a pruning avoidance. So we wanted to look at, does it have an effect on bud development here in our region, but also does it have an effect on any of the berry characteristics, fruit characteristics. So now this is from last year, and again, we were lucky that we got a specialty crop block grant funded which allows us to continue this research and even start doing more of this research. And that is the same team. It's myself, it's Sarah, it's Emma, and Rachel Kreis. And our all goals here are to do a comprehensive survey of pathogens in North Carolina. We're actually doing it right now. Emma was sampling a lot of wood in the last couple of weeks. And we also developed pruning wood management strategies in North Carolina. We have 2 large trials with Vidicil and Thompson M and Raleigh, and different pruning strategies here in Hendersonville. And then also we develop molecular identification tools for the PDSC next year. So that is kind of our plan. For now, I am on my way right now in the car to go to this field trial because we're going to implement our treatments this weekend. So now, if you have disease symptoms and you do not know what those disease symptoms are, because sometimes it's very clear, but most of the time it is not very clear at all, and you only have some redixing and chlorosis, I don't want you to run directly to the PDSC and say, oh, I have a virus. And I also don't want you to run directly into PDSC and say I have trunk disease. Virtually, you will have trunk disease in any vineyard in North Carolina. We didn't find any vineyard without trunk disease so far. But it's not always a big problem. We want you to go systematically on your problem. So if you have reddening and chlorosis, or you have crisp leaves and you do not know what that might be, your first guess should be, okay, it might be something which is nutrition related. You take your paleo samples, take your soil samples, talk to your nutrition specialist, see and figure it out. If there's nothing wrong with your nutrition and you're going to be okay on your nutrition, then you would check for physical damage on the wine. You can do it at the same time. See if you have any physical cracks from old cold damage often, or from any other physical injury, which might lead to this damage. If you do have physical damage, you might still want to look for different diseases. If you don't find any crisp leaves or on your wine, you probably don't have to test for Pierce's disease. I would still go look for crown gall and for trunk disease in your wine. If you do have crisp leaves, I would throw in some Pierce's disease. Pierce's disease is very clear symptomatically, but sometimes it's not. And those are the times where you would basically go through this whole system. If any of this is negative, so you don't have Pierce's, it's not trunk, it's not crown, don't see a lot of physical damage, your nutrition is fine, that is when you would say, you know what, this looks very much like a virus. We could exclude everything else. So now let's test for a virus. And that is also a reason why you would send that sample separately, because it has to be on ice, and it has to be cooled, and it has to be shipped within 1 day, and it has to be sterile. And so my time is way more than over. So we do have a couple of other research projects that is finished. One which I really would like to highlight is we did a larger study in collaboration with Kane Hickey and Eric Case and Annie Fogel here at Burnshot again on leaf removal strategies. So that study is over. We did our last harvest last year. And we wrote a publication, an extension publication, quite really nice extension publication on it. And that should be out before you have to do your leaf removal. So stay put on this one. I hope we can get this out in the next couple of weeks. Also, new research projects that will start, with the exception of the trunk disease. In addition to trunk diseases, we were starting a cultivar trial at Childress Vineyards for Pierce's disease-resistant cultivars. That includes the UC Davis cultivars there. So we're going to plant that trial this year. And then we also have a project to optimize light impact on flower bud development the year before flowering. And looking in strategies there, and that is in a larger collaborative effort, which also starts this year with at our NC State. And with this, some of the resources which you should follow, and then I am there for questions right after this. And thank you very much.